Welcome everyone for the SWAM course on genetic engineering and its recent advancement. I, Dr. Sharmishta Banerjee, working as Assistant Professor in Chhattisgarh Swami Vivekanand Technical University, will be discussing today restriction endonucleases and its types. Let's engage in this 30-minute video lecture on the topic. In the last few lectures, we have learned how do we isolate the desired gene of interest, or we can say transgene, how it is introduced into the host cells. In this context, restriction endonucleases play a very important role as it is the most important tool without which we cannot proceed further. So, in this lecture, we will explore the fascinating world of restriction endonucleases, also known as restriction enzymes, and we will delve into their various types and functions. So, what is a restriction enzyme? It is a nuclease. What do you mean by nuclease? The enzymes which are capable of degrading nucleic acids, basically DNA or RNA. So, it is a nuclease enzyme that cleaves the DNA sequence at random or specific recognition sites, which are majorly referred as restriction sites. It is a protein that recognizes a very short stretch of nucleotide sequence of DNA, and which is basically our restriction site or target sequence or recognition sequence. It first recognizes and then cuts this DNA only at that specific site. These enzymes are majorly found in bacteria as well as some other prokaryotic organisms. Each restriction enzymes recognizes just one of few restriction sites. It can be either a single restriction site in a DNA sequence or RNA sequence, or it can be multiple restriction sites. So, after recognizing its target sequence, a restriction enzymes makes a double strand cut in the DNA molecule. Typically, the cut is at the restriction site or near to it. It will count some base pairs from the target sequence and then it will produce a cut in some cases. And these cuts, they occur in a very orderly or predictable pattern. So, in bacteria, these restriction enzymes, they form a combined system which is known as restriction modification system. And in addition to these restriction enzymes, it also have modification enzymes. For what are these needed? They are basically needed for methylating the bacterial DNA. In detail, we will study this topic in further lectures. Methylation of bacterial DNA at the recognition sequence typically protects the own DNA of the bacteria from being cleaved by restriction enzymes. What do I mean by this? I will explain in the figure in the upcoming slides. Basically, from the name itself, restriction enzymes, they are restricted for a particular site, and these restriction enzymes are majorly classified into two: exonucleases and endonucleases. Already I had given you the glimpses of these, but just to have a quick recap, I will again repeat the definition of these two restriction enzymes. The first one is exonucleases. From the name itself, it will cut at the ends of the DNA or RNA molecule. It can be either at the 5' end or at the 3' end. So, it catalyzes the hydrolysis of terminal nucleotides from the end of DNA or RNA molecules. The examples of these are exonucleases 1 and exonucleases 2, and there can also be other examples of it. Second category of restriction enzymes are endonucleases. From the name itself, endo means within. It recognizes the specific base sequences within the DNA or the RNA molecule. These base sequences, I told you, they can be called as restriction sites, target sites or recognition sites. I am repeating again and again so that you get familiar with these terms. So, these endonucleases have been recognized by a specific base sequence, and they will cleave the internal phosphodiester bonds that are present within a DNA molecule. Basically, these bonds are capable of joining the two nucleotides in a nucleic acid chain in a DNA or a RNA molecule, we can say. The examples of endonucleases are EcoR1, Hint3, BAMH1. And today in this lecture, I will majorly focus on endonuclease, which are most important tool of genetic engineering or of our subject, I can say. Here in this figure, we can see, in the first figure, we can see that this is the basically endonucleases. It will recognize a site, and we can see that the two strands are getting separated from within, or a particular segment of DNA is being cut from the whole set of DNA molecule, from the whole strand of DNA molecule. So, in this way we can select our transgene, we can cut our transgene from the whole genome, or we can even produce a cut in the genomic DNA to have this transgene inserted at a particular site, or we can even produce nick in the vector molecule to produce a recombinant molecule. So, for that reason, since this is a tool which is used for cutting the DNA strands or for cutting or isolating the gene of interest, that is why they can also be termed as molecular scissors. So, the question is how these restriction endonucleases were discovered. So, the initial observation was made early in 1950s, when it was shown that some bacteria are immune to bacteriophage infection. Why? This is basically governed by host controlled restriction system, or we can say restriction modification system. In detail, we will study this particular topic in the next lecture, but I will just give you a glimpse of it. So, what is a bacteriophage? Bacteriophage are the viruses that infects a bacteria. So, in this figure, we can see the first half of the figure, or the A part of the figure. What we can see, the diamond shape, or basically it is a bacteriophage molecule that is latching in the host DNA molecule, or we can say that is being latched upon the host cell. So, this foreign genome or foreign DNA or RNA from the bacteriophage is being inserted into the host cell, and what we can see, this host cell, they are not being cleaved, or it is getting protected. Why? Because of the methylation pattern, and the explanation we will study in detail in the next lecture. So, understanding the biological basis of host controlled restriction and modification of bacteriophage DNA led to the identification of restriction endonucleases. That is the question, why this particular host cell was immune to the bacteriophage. It led to the discovery of the restriction endonucleases, and it was observed that these bacteria, they have restriction endonucleases, and that is why they are cleaving the foreign genomic material when it is entering into the host cell, and in this way they are getting non-functional or dysfunctional and cannot have a cycle of infection in the host cell. So, till date, more than 400 restriction enzymes have been isolated from the bacteria that manufacture them. In live bacteria, restriction enzyme function to defend the cell against invading viral bacteriophages. Restriction enzymes of the bacteria cleave the restriction site in the viral genome, fragmenting and destroying the DNA of invading bacteriophages before it can even incorporate into the host bacterial genome and take over the cell to an infection. Interestingly, a bacterium is immune or resistant to its own restriction enzymes. Again, this question will be answered in the next lecture in restriction modification system. And the most surprising thing about this is, even the bacterium contains the target sequence of a particular restriction enzyme, even though it is immune or it is resistant to its own restriction endonucleases. The nature of cleavage produced by a restriction endonuclease is of considerable importance. Now, we will discuss what is the pattern by which the restriction endonucleases creates cut in the DNA molecule, and here we can see that they can be categorized, or the DNA molecule is basically cut in two different ways, which are as follows. The first one is many restriction endonucleases cleave both the strands of the DNA simply at the same point within the recognition sequence. What do you mean by this? I will explain it in figure. As a result, this type of cleavage, the DNA fragments will produce blunt end. The example of such type of enzymes are PVU2, HAE3, ALU1, which produces blunt ends. Now, the question is, what are blunt ended DNA molecules? Blunt end may also be referred as the non-protruding ends. Here in this figure we can see the double stranded DNA molecule. In both the figure, in the first half of the figure we can see that the recognition site is in between in both the figures, but in the first figure what we can see that we have overhanging ends. We have the two strands separated in a sticky pattern. So, in the first half of the figure there was an enzyme which was producing cut in such a manner that the ends are not even, the ends are protruding. But in the second case what we can see that no overhangs are there, no protruding ends are there, no sticky ends are there, and they are basically generating blunt ended DNA molecules. In the other style of cleavage by the restriction endonucleases, just we saw in the previous slide, the two strands of DNA are basically cut at two different points. Such cuts are termed as staggered cuts, and this results into the generation of protruding ends. That is, one strand of double helix extends a few bases beyond the other strand. Such strands are also called as cohesive or sticky ends. Here we can see the example of an sticky end cutter as well as a blunt end cutter. So, in the first half of the figure or upper half of the figure, what we can say, ECO R1 was the restriction endonucleases, and in both the cases you can see the target DNA molecule was same, but the restriction sites or the recognition sites in the upper half was GAA-TTC, but in second case it was GATA-TCC. So, what we can see in the first case that the enzyme cut the DNA molecule after the first G, and in the upper strand of the DNA, that is which is running from 5' to 3'. So, again these cutters, or you can say these recognition sequences in both the strand, when you see that it reads the same way. In the upper strand also it is GAA-TTC, in the second strand also, that is in the strand of 3' to 5', the enzyme sequence, restriction sequence is GAA-TTC. So, the arrow is basically indicating that the DNA molecule will be cut after the arrow as soon as it recognizes the recognition site. So, we can see where is the arrow? The arrow is present after G in both the strands. So, let us imagine you do not see the second half of the figure. Let us see how after G the cuts will be produced. You can even draw it with the help of pen and pencil on a piece of paper. So, we can see that the ends which are created is sticky when it is being cut by ECO R1. You can see in the first half, that is ATGCG, that is in the 5' to 3' direction of the DNA. So, the upper strand of the DNA is getting ended there itself, but in the second case we have a staggered. So, that is basically a sticky end cutter, how it works. I hope it is now clear to all of you. Then in the second half of the figure we can see ECO RV was the enzyme, and the cut was produced uniformly at the same site for both the strands, that is for 5' to 3' as well as for 3' to 5' strand of the DNA. So, what you can see, there are no overhangs, blunt ends are being produced. Now, Basically, we can divide the restriction endonucleases or restriction endonucleases in three different categories: type 1, type 2 and type 3. Now, what are type 1 restriction enzymes? They recognizes specific DNA sequence, but cut at random positions away from the recognition site. It's composed of three different subunits. The first one is recognition, second one is modification, and the third one is restriction. So, there are three different subunits of a single enzyme, and this is how it works. It is also an active process. Why? Because it requires ATP for their activity. The type 2 class of restriction enzymes, it recognizes specific DNA sequences and cleaves within or near the recognition sequence. Most commonly, this is the type which is used in molecular biology and genetic engineering. The examples include EcoR1, BAMH1, HINT3, and this process has an advantage that it does not require any energy or ATP molecules for their activation or for completion of this particular type of digestion. Restriction sites in double stranded DNA have a two-fold axis of symmetry, which is basically known as palindrome. Just now we have observed that in both the strand, when we are observing the DNA strand or the recognition site, it reads the same way from the left to right as well as from right to left also. So, these are basically palindrome. I will give you an example which will make you more clear. Like the word madam, if you spell it from left to right, it is MADAM. Similarly, if you observe it from right to left, it is again the same. So, I hope it is clear that this restriction enzymes, they are basically having their recognition sites as palindromic sequences. So, that is why they will fall a two-fold axis of symmetry. Cleavage can leave staggered or sticky ends or can produce blunt ends. The type 3 restriction enzymes, they are very rarely used, but to give you a knowledge, I am just giving you an quick glimpse of it. It recognizes the specific DNA sequences. It produces cut at a variable distance from the recognition site, and it also have a modification subunit. 4 different types of restriction enzymes have been shown in this particular table. What we can see is type 1, type 2, type 3, and all these 3 we have already discussed, and we can see that in the first case I already told you that the recognition site as well as the restriction sites, they are different. So, here also we can see it cleaves around some base pairs away from the recognition site. In type 2 they are the same, in type 3 again it is different and it is 24 to 26 base pairs away. Whereas, what is type 2S? A type 2S are acting on those and it produces asymmetric cut, we can say. Cleavage can occur at the recognition sequences up to 20 base pairs away. These are the examples of the restriction endonucleases along with their recognition sequences, that is SMA1, HINT3, EcoR1, all these are denoted here in the table. So, in the further slide I will tell you how they are named as such, or what is the principle behind their nomenclature. The naming of this restriction enzymes, they provide the detail about their sources. So, again coming back to the principle, because type 2 is the most commonly used, so I will discuss in detail a bit of it. So, type 2 restriction enzymes, they cut DNA at specific nucleotide sequences. A particular enzyme cleaves DNA at its recognition sequences and nowhere else. Already the examples I have discussed, but here I will give you a quick detail about some of the enzymes. One is PVU1 or PVU3. From the name itself, both have been isolated from the same source, that is Proteus vulgaris. They cut hexanucleotide sequence. What do you mean by hexanucleotide? Basically, they are 6 base pair cutters. Like if we count the bases in this recognition sites, that is CGA, TCG, these are 6 in number. So, this is a hexanucleotide cutter. Many restriction enzymes, they recognize hexanucleotide target sites, but there are some which can even produce cut at 4, 5, tetra, penta, octa or longer nucleotide sequences. They all have a two-fold axis of symmetry. That means basically they are palindromic in nature. There are also restriction endonucleases which degenerate recognition sequence. Example is in F1, they recognizes GANTC. What do you mean by degenerate nucleotide sequence, degenerate recognition sequence? Like we have GANTC. So, all the other base pairs are clear in this particular recognition sequence except N. N is any nucleotide. So, I have elaborated what is this N. N can be A, T, G as well as C. So, they have 4 different types of recognition sequence. The same enzyme have 4 different type of recognition sequence as we can see. That is, whenever in the DNA sequence it finds GAATTC, it will produce cut, GATTC, GAGTC and GACTC. It is simpler. In place of N, if any nucleotides are present, it will produce cut. Now, the question is how these restriction endonucleases work. What is the principle behind their cutting mechanism, principle behind their digestion? They recognize specific DNA sequence, they cleave DNA near the recognition site, resulting in blunt or sticky ends. As an example, how a restriction enzyme recognizes and cuts a DNA sequence, let us see an example of EcoR1, which is a common restriction enzyme which is used in biotechnology labs. The EcoR1 cuts the following restriction site. Already the restriction site, here I will say restriction site because in case of type 2, that is the recognition as well as the restriction sites both are same. So, what is the EcoR1 restriction site? Already we have seen in the previous slide. It is GAATTC, which is being highlighted in purple in this particular figure, and I will show you that these are palindromes. Just read it with me. In the upper strand it is GAATTC. Since we have a complementary other strand, again we can see in the lowest complementary strand that the C will correspond to G, T will correspond to A will correspond to T and again G will correspond to C. So, read the lower strand of the restriction site, it is again GAATTC. So, these are palindromes and they will follow a two-fold axis of symmetry. If we place these two DNA strands together in a straight line, they will appear as mirror images of one another. Just see. So, what is a palindrome? Now it is clear, but I will just give you the definition of it. It is a nucleotide acid sequence on double stranded DNA or RNA, wherein reading it from 5' end to 3' end forward on one strand matches the sequence reading from 3' end to 5' end on the complementary strand, within which it forms a double helix. The top strand reads, already I told you, it reads from 5' to 3' as 5 to 3' as GAATTC, while again the bottom will read the same. If the DNA strand is flipped over, the sequence are exactly the same. An EcoR1 binds to an EcoR1 site in a piece of DNA and makes a cut on both strands of the DNA, and it is shown here in the diagram. The arrow indicates the cut site. When EcoR1 recognizes and cuts its restriction site, it always do so in a very specific pattern that produces ends with single stranded overhangs. Thus, it produces an overhang of, if it will produce a cut after G, what is the overhang that is present? It is AATTC on 5' to 3', but the C is absent. Why? You can see that it is complementary. So, if again it produces a cut on the lower half of the DNA strand, then the C will be absent. So, overhang will be till T only. The pattern of the cut is called as staggered cut since it produces sticky ends. and it is already shown to you. EcoR1, here again we can see the how the sticky ends are produced, already I had explained to you. So this is how the restriction enzyme is actually working. We can see in the first half of the figure, or in the first step, the black spherical balls, they are basically restriction enzymes. It is coming and binding to the recognition sequence, that is EcoR1 enzyme was coming and binding to the recognition sequence which was present in the double stranded DNA molecule, and it will target the recognition sequence, produce cut in the target sequence, and in this way the overhangs are produced. Later on, to join this with the other DNA molecule, either it can be a vector molecule or the host genomic DNA molecule, it will be joined with the help of ligase enzyme according to our need, and a recombinant DNA molecule can be constructed in this manner. The two sticky ends, they are joined together by complementary base pairing. If another piece of DNA has matching overhangs, again this joining is not random, it will only join if it has matching overhangs, and it can occur when both DNAs will be cut by using the same restriction enzymes. The overhangs of the two different DNA stretches can also stick together by complementary base pairing. For this reason, enzymes that leave single stranded overhangs are said to be producing highly reactive sticky ends on the broken ends. Again, it is a disadvantage, or we can say it is a challenge we have to overcome in most of the cases. Sticky ends are helpful in cloning and genetic engineering because they hold two pieces of DNA together, so they can be linked by DNA ligases. Not all restriction enzymes produce sticky ends, some also produces blunt ends. We had already seen the example of such enzyme is SMA1. SMA1 enzyme binds to the SMA restriction site and it is given below. Again, it will make a cut right in the middle of the sequence on both the strand. Since it is a hexa cutter, so after 3 nucleotide in the recognition site, it is producing cut, that is CCC. After CCC, a cut is being produced in both the strand, so blunt ends will be generated. However, these blunt end fragments, they are very harder to ligate or join together. Why? Because the ligation reaction is less efficient and it is more likely to fail in this particular case, because there are no overhangs to hold the DNA molecules in their specific position. Restriction enzymes with the same specificity are known as isosizomers. So, here are some examples of it, like EcoR1. We can see what is the restriction site of it is GAA-TTC. Isosizomers I will discuss in further slides. So, here I can tell you that either the sticky ends or blunt ends are produced with the help of these recognition sequences, with the help of these restriction enzymes. Applications: they are used in the process of insertion of genes into the plasmid vectors during gene cloning and protein expression experiments. Restriction enzymes can also be used to distinguish gene alleles by specifically recognizing the single base sequences in DNA, which are basically known as SNPs or single nucleotide polymorphism. This is only possible if a mutation alters the restriction site present in the allele. Restriction enzymes are used for again the marker analysis, or we can say more specifically RFLP analysis, that is restriction fragment length analysis, length polymorphism analysis, for identifying individuals or strains of a particular species. It can also be used in DNA cloning and DNA mapping. Let us have a quick walk through the process of DNA cloning using restriction enzymes. Target DNA and the vector molecules will be isolated and both will be digested by using the same restriction enzymes. Here, in case of DNA mapping, what we can observe is the how much base pair long our DNA interest DNA is. So, with the help of this restriction endonucleases, a specific DNA site can be mapped, its length can be predicted. Map means the exact location of that particular gene can be obtained, or it is also known as gene mapping with the help of restriction endonucleases. Let us discuss few more points on gene mapping, how the restriction endonucleases are helpful in mapping the genes. So, what is gene mapping? I had already give you a brief about it. It basically determines the order of genes and the relative distance between them in map units, and these map units are most commonly termed as centimorgan, named after the scientist Morgan who discovered this particular technique. And one map unit is equal to 1 centimorgan, which is abbreviated as small c capital M. Please do not confuse it with centimeter. Okay. Gene mapping method use recombinant frequencies between alleles in order to determine the relative distances between them. So, the question is, what are alleles? Alleles are a pair of contrasting characters which are present in a gene. Contrasting character means like the if a gene is capable of indicating the height of a particular organism, there can be a tall allele as well as a dwarf allele. These are the two features. So, by allele what I mean is this: recombination frequencies between genes are inversely proportional to their distance apart. So, if one map unit is being shown in the map, that means there is 1% recombination, and this definition holds true only when we are taking short distances. It can also give us an idea of genetic linkage. Genes with recombination frequencies which are less than 50%, we are said it to be a linked gene because they are present on the same chromosome. Linkage group means all known genes on a chromosome. Two genes that undergo independent assortment have recombination frequency of 50% and are located in non-homologous chromosomes, or they are far apart on the same chromosome. If they are present on the same chromosome, they will be far apart. So, they are basically termed as unlinked genes. Now, how these RFLP analysis is basically done with the help of restriction endonucleases. We will just have a quick recap of it. Differences in DNA sequence generate different recognition sequences and DNA cleavage sites for specific restriction enzymes. Two different genes will produce DNA fragment patterns when cut with the same restriction enzyme due to difference in DNA sequence or due to the presence of the restriction site at a different location in two different DNA. That is why this analysis will help in identifying different strains, different individuals or different genes, we can say. The nomenclature is how it is being, every restriction endonucleases is named. So, first the restriction enzymes was isolated from the bacteria, and the nomenclature system depends upon the type of the bacteria from which the enzyme has been isolated. Let us take an example to see the nomenclature system. Again the most common example which I will be taking is EcoR1. So, EcoR1, the first letter is always written in capital letter like E in this case, where it represents the genus of the bacteria from which the restriction endonuclease has been isolated. So, E stands here for Escherichia. The second two letters, that is small c and O, again it will be written as in small letter. The second two letters, it represents the species of the bacteria, which is coli in this case and is written as Co. The third letter is again designated in capital letter, that is R, and it represents the strain of the bacteria and it varies with different species. Again it is followed by a Roman numeral, that is 1 in this case. What does it indicate? The number 1 is signifying the order of identification of the particular enzyme. So, Let's have now a quick recap of what we have studied till now. What are restriction endonucleases, its definition, introduction to it, how they can be isolated, where they are present, all this we have discussed. So, what are endonucleases? They are basically nucleases, or they are proteins, or they are enzymes which are producing cut in the nucleotides. How they work? Already we have seen. They will identify the recognition sequence and produce cut at the restriction sites. The types, basically they are categorized into 3 types: type 1, type 2 and type 3. Out of them, type 2 is the most commonly used. Application of restriction endonucleases we have studied in SNP analysis, RFLP analysis, in cloning, in DNA mapping, and there are numerous applications of these restriction endonucleases. What is the basis of nomenclature of these restriction endonucleases we have discussed here. So, in the next lecture we will learn the restriction modification system in detail. Thank you for joining us.